Now, we would like to begin Dr. Yoshinori Osumi's lecture. Dr. Yuko Fujioka from Hokkaido University will be the moderator of this session. Please welcome Dr. Fujioka.
Good morning, everyone. I'm Yuko Fujioka from Hokkaido University. It is my great pleasure to moderate this session. I'd like to briefly introduce Professor Yoshinori Osumi. He was born in 1945 in Fukuoka, Japan, and he began his scientific journey in 1963 at the University of Tokyo. After obtaining his PhD from the same university in 1974, he studied at the Edelman's Lab at Rockefeller University, USA, where he had the opportunity to study yeast systems for the last year. In 1988, upon returning to Japan, he discovered autophagy in yeast and has been involved in its research for over 35 years. Currently, he runs a laboratory as an honorary professor at the Tokyo Institute of Technology. Now, I'd like to invite Professor Osumi to give a breathtaking talk. Dr. Osumi, please start when you're ready.
Thank you, Yuko. She's my long-time colleague in autophagy research. Okay, it's a great pleasure for me to be here as a speaker of this 15th HOPE meeting. Today, I'm already studying problem of autophagy already 35 years. Just using yeast cell. Now, autophagy has become really one of the most popular fields in biology, and expectation for application is now expanding. Today, I will briefly introduce you what is the protein degradation, how I launched into the autophagy field in using yeast cell, and briefly recent our new data on autophagy. And finally, I want to discuss with you about the basic science in all over the world and my hope for next generation. When I was a graduate student, central dogma has been establishing. Fascinating by this existence of a principle common to all life on the earth. I want to be a molecular biologist in future. But on central dogma doesn't say about the protein degradation. This simple diagram is a daily protein dynamics in our body. Nutrient study tells us about 70 grams of protein you should eat, take from diet every day. But our body are making about 200 to 300 grams of protein every day. Just these number figures tells us most of the protein we need to live is made by the amino acid degraded by our own protein. So for synthesis, degradation is essential. So this quite simple figure tells us our body life is quite a good recycling system. So in summary, life is maintained equilibrium of the protein synthesis and degradation. So every 3 months, almost all protein in our body is replaced for new protein. That's a way of life. And how the recycling is important in our life. Okay, one severe stress for life is starvation. If we can live just water for 1 week with just on water, that means that all of the required protein synthesis is made by recycling. So acquisition of the recycling must be very important for the evolution of the life on the earth. So autophagy is a major recycling system and play critical roles in evolution. However, studies on the intracellular protein degradation was thought to be interesting, but the progress was so slow because many people thought that protein is quite stable in a cell, in our body. And also, degradation is a passive process, not active. So not so many people interested in the degradation process. In 1955, Christian Dudu, Belgian cell biologist, found the lysosome, single membrane bound organelle containing various kinds of hydrolytic enzymes inside, acidic. So people thought dangerous degradation should be occurring within this membrane. So many people at Rockefeller University were electron microscopists work on the process by electron microscopy and found cytoplasmic protein is how incorporated in the lysosome. And Christian Dudu coined the process in 1962, autophagy. Autophagy auto means a self, phagy means eating, so Greek, self-eating in Greek. So autophagy has a quite long history, but no genes or no protein involved in this process has long time, no information about the kind of things. But this slide shows the mouse EL cell starved 2 hours, amino acid starvation. As you see, these black structures are autophagolysosome, just degrading cytoplasmic component within lysosome. So autophagy is a quite popular phenomena in our body. If you feel hungry, between meal already liver cell, you can have liver cell start autophagy. So it's quite popular phenomena, but not much progress was done about autophagy. Okay, from here I will talk my studies. I was working on the yeast cell. Yeast is a quite tiny, 5 micrometer small unicellular organism. But as you know, you eat bread, you eat some yeast cell. So yeast is a quite important organism, useful organism for fermentation and baking. But also yeast cell is a very good model system of eukaryotic cell. So genetics, molecular biology, cell biological methods are well established in this field, and also genome sequence was established in this organ. We have so many data about this on small organisms. So very good system. When, as you got introduced, when I came back from the United States, I started yeast work at the University of Tokyo. My boss told me, you can work any subject on using this yeast cell. So that's a starting point of my yeast work. After much deliberation, I decided to work on this round structure, vacuole. Vacuole was just thought as a garbage dump of the cell. So not so many people interested in this structure. But always my principle, I want to work the subject which is not popular and not so many people interested in. That's my principle in science. But as you think, plant cell has a very huge vacuole, contains 90% of the cell volume is a vacuole. So vacuole should be very important for plant life. And there must be various kind of function on this organelle. That's the reason I want to first cut this organelle. So I prepared the vacuole, purified vacuole, and vacuolar membrane vesicles, and I found vacuole has quite many active transport systems on it. And driving force is given the V-type ATPase. That's a proton pump, Nobel proton pump. That is very important for cell biology. That was first found by our groups. So actually, vacuole is quite active and important organelle. However, I worked 10 years about the physiology of this organelle, but in 1988, turning point came to me. I had my own small lab just by myself, so I want to work another subject. That was a vacuole, might be a lytic compartment homologous to lysosome in yeast cell, because vacuole is acidic, and it was known that vacuole contains various kinds of hydrolytic enzymes in it. If so, the problem should be many cytoplasmic protein, how those are come across the vacuolar membrane to meet the degradation enzymes within the vacuole. That's the original question I had. So that must be a membrane phenomena. Might not be so easy, but that I thought that is an important question. When is cell do massive protein degradation? I was interested in the sporulation process in yeast. When yeast cell face to the nitrogen starvation, diploid cell form 4 spores shown here. So this quite dynamic remodeling of the cell within cell is occur without any supply of the nutrient. So this process should occur massive on protein degradation. So nitrogen starvation might be a very important condition to interesting condition to work with. And another important point is I like light microscopy. The yeast vacuole is the only organelle you can see under just a light microscope. It's large, and also vacuole is just almost just a solid solution. So viscosity is so small. So I knew every day I observed the yeast cell. So I knew some structure get into the vacuole. I can easily see under light microscope. So my starting point is I might see some transport of the material to the vacuole might be seen under microscope. So I carefully observed the morphology of the vacuole under nitrogen starvation condition, but I couldn't see any change. One day, one idea came to me: if I use a protein, vacuolar protein deficient cell, that might keep the material within vacuole delivered, is detained. So I could see some change during starvation. This movie is just I found at the moment. As you see in the light movies, many spherical structure moving around in the vacuole. So I thought I faced quite interesting phenomena. That was just a starting point of my autophagy research. This is the time course of the phenomena. Even after 30 minutes, this kind of black dot structure appeared in the vacuole. And as you saw, within 30 minutes, almost vacuole is filled with this kind of structure. So we studied the electron microscopic studies on this phenomena. The first, this is a structure within the vacuole. We named autophagic bodies. Those are single membrane bound structure, and this black dot is a ribosome. So you can see the same density of ribosome within autophagic body. That means a portion of cytosol is taken up by a single membrane. Those are final stage of the delivered to the vacuole. So the obvious next question is how these kind of things happen. So again, electron microscopy tells us a lot. First, just next to the vacuole, the small membrane sac appear and elongate and form this kind of cup-shaped structure, and finally form a double membrane structure, autophagosome in the yeast. And this double membrane structure is stick to the vacuolar membrane, and outer membrane of vacuole fused with a vacuolar membrane. You see the inner membrane structure just showing. And these structure are we call the autophagic body. It's a structure you saw in the moving around in the movie. So, and also this movie showed the autophagic membrane is quite unique biomembrane. There is no protein particles on this membrane. And the reason I show this slide is autophagic degradation is characteristic to degrade so many ribosomes, more than 1,000 ribosomes. Ribosomes are quite large ultrastructure, but those many ribosomes are taken up one autophagosome. And as you see, mitochondria is taken up in those kind of membrane structure. So that is a quite unique degradation system to degrade bulk cytoplasmic structure in the vacuole. So more than 30 years ago, we could draw this kind of picture to explain their autophagy in yeast. When yeast cell face a starvation condition, not only nitrogen source, carbon, sulfate, any kind of phosphate, and zinc, many kind of starvation condition, yeast cell will suddenly start formation of the small membrane structure and form a double membrane structure, autophagosome, and inner membrane structure is fused in outer membrane structure, vacuolar membrane, and inner membrane structure is taken up to the released to the vacuolar lumen. The point I found this phenomena is that I used the vacuolar proteinase deficient mutant cell. So in wild type cell, this structure is immediately disrupted to reuse their contents in wild type cell. So nobody found this phenomenon. But I used the proteinase deficient mutant cell. I could follow their autophagy in light microscope in real time. That's a good important point for us. Just recently, we realized why vacuolar protein deficient mutant cell behave this kind of, show this kind of phenotype. The reason is lipase is important for degradation of the membrane, but lipase, ATG15, should be activated to work to disrupt the membrane. So autophagic vacuole deficient mutant cell cannot activate the lipase. So that's the reason we see the accumulation of autophagic body we found recently. So next problem for us is a genetic approach to this phenomena to dissect one by one. So, but we didn't know anything about the phenotype of autophagy defect mutant cell. So we started just a morphological study under microscope, one by one, find the cell which cannot accumulate autophagic body in the vacuole. And fortunately, my first graduate student, Miki Tsukada, found first autophagy defect mutant, APG1-1, that now we rename ATG1 mutant. But that process should have many more genes, so we started to isolate other ATG genes by using one of the phenotype, ATG1 cannot survive under starvation conditions. So we first correct the loss of viability phenotype. Then under microscope, we found 15 ATG defect mutant. And finally, we add 3 mutant. So finally, we found 18 ATG genes are essential for the yeast autophagy. Surprisingly, all these genes are Nobel genes. No information about their function. First, it was a very difficult time for us, but that was the reason most of the genes are analyzed in even so many people working in yeast cell. And ATG genes is function as a complex, so it was very difficult to understand the real function of each ATG genes. But fortunately, I moved to the NIBB. Our group became bigger, and within short time, we found 15 ATG genes are divided into this kind of 6 functional groups. I don't want to go in detail. One is a protein kinase and the lipid kinase complex, and interestingly, 2 ubiquitin-like conjugation system and ATG9 membrane protein and ATG218. I will briefly go back to this protein. And also, Fujiko Inagaki, Takeshi Noda, Yukiko belongs to their groups. We started a long-time collaboration about the structure biological study of the ATG genes, ATG protein. And finally, we got many information about the ATG protein structure. That helped a lot to understand the detail of the autophagy gene function. Next question is how and where ATG protein are working. GFP was found by that time ATG was found, so that was a great tool to understand this problem. And this slide shows one of the examples, ATG17. ATG17 is just moving around by Brownian motion and on the side. So, but if starvation occurs, some of the ATG17 accumulate in the one dot structure shown, white dot structure. So I thought that might be the autophagosomal malformation site. We called it a path pre-autophagosomal structure. And those kind of analysis after we drew this kind of overview of the sequential uptake or sequestration of ATG proteins subcomplex. First, ATG1 protein kinase complex come to the path. And then ATG9 comes and PI3 kinase join, and 218 complex join, and finally 2 conjugation system works. And I will just mention about the autophagic induction of the autophagic process. We know that ATG17 is a key molecule that is highly phosphorylated under growing condition. Starvation condition, those phosphate are deleted, and dephosphorylated ATG formation is a signal to start the autophagy. ATG17 interact with ATG17, ATG1, and form a pentameric complex. And those pentameric complex formation is not sufficient to induce autophagy. They should have a more bigger structure. We didn't know about those kind of structure, but quite recently, Yuko and Noda's group solved this kind of problem, and this protein in the cytosol accumulated the one-dot structure path. That is a so-called liquid phase separation. So autophagic machinery itself is formed by this kind of liquid protein interaction. Separation step is very important to form an active path. And also, so early path is formed. ATG1 is recruited in this structure. ATG1 kinase is phosphorylated and activated. That's the starting point of the next step of autophagic membrane formation. 87 vesicles, small number of vesicles form a seed membrane, and the elongation steps occur to form a membrane structure. Long time we didn't know where lipid molecule is come from the isolation membrane. That problem was also solved in the Noda's group recently, and ATG2, 18 complex, is very important for this process. ATG2 is a lipid transfer protein from the donor membrane to isolation membrane. And also this membrane is scrambled to the inner membrane, reflected by ATG9. That's a reason, that's a mechanism of autophagic membrane expansion. As I said, we established 18 ATG gene in yeast. Now in NIBB, our group showed almost all ATG genes are well conserved from mammals and plants, shown here. So actually, identification of ATG genes completely changed the autophagic research landscape. One example is if you put the GFP to one of the ATG genes in mammals and express in systemically, shown here, Nobel Mizishima made a GFP LCC mouse, shown here. But using this mouse, you can easily see under fluorescent microscope what tissue, muscle, liver cell, even fertilized egg. These white structure, dot structure is autophagosome. And the fibroblast taking up those kind of mouse fibroblast is starved only 1 hour, this kind you can see many white dots. Those are autophagosome. So many autophagosomes are formed under starvation conditions shown here. And also, as we made the autophagic defective mutant, now we can make autophagic gene knockout mouse. And first autophagy knockout mouse was made Nobel Mizushima by ATG 5 knockout, double knockout mouse. We didn't know it can survive, but they double knockout strain can you show that you see 100% alive, but within 12 hours, those mouse can die. So autophagy is essential for growth of their autophagy. Cannot survive under mouse cannot survive after birth without starvation. So many labs started tissue or organ-specific knockout of ATG genes. One example is the Komatsu group showed the knockout, ATG7 knockout in liver cell. You can see this kind of large, enlarged liver, and those liver cells contains many ubiquitin conjugated protein aggregate within cytosol, and those mouse behave like neurodegenerative phenotype. And just starvation is mammary cell. No neuronal cell is protected by starvation, but that doesn't mean autophagy is not essential for neuronal cell. Actually, ATG knockout cell in neuronal cell shows this kind of aggregate protein deposit within cell and shows neuronal defect. And also, Tamotsu Yoshinori's group found autophagy is essential to kill the invasive bacteria within cell. And this process, as you see here, the magenta shows the bacteria is quite specifically adapt to the autophagosome membrane. So this is a very good example of the selective autophagy. One target is specifically enwrapped by the membrane to be degraded. So this is now many people started the selective type of autophagy. That is one of the good, nice topic to work with. So after ATG gene, many labs all over the world worked on the autophagy in various cells, various organisms, and found autophagy is essential for nutrients recycling, but also clearance of the cytosol degraded, unwanted or various kind of material from the cytosol, including bacteria, shown here. And also, autophagy is very important for the early development of the mouse. Many people interested in the autophagy-related longevity. Many people asking questions on this problem. And also, now, cancer cell requires autophagy for good proliferation. So many pharmaceutical company want to have a specific drug to control the autophagy to cure the cancer cell. So this slide shows the transition of the paper related to autophagy. As I said, I started in ATG, autophagy study 1988. That time only 20 papers appeared per cell. Now, these days, more than 10,000 papers appeared 1 year about the autophagy. So I feel this kind of burst occurred just our quite small finding. I'm very happy. And I want to also to say two things. If autophagy was progressed quite nicely, but it took 30 years to become this kind of peak. So basic science requires some long time to continue. And another thing is many young people in Japan want to work on the popular subject. So many people interested in the autophagy right now. But I dare want to say some people challenge to the new subject. Some here, not so much interest right now, but make this kind of peak in near future. So I have chance to move to the yeast cell to mammalian cell, but I feel my mission in autophagy research is just answers the first question: what is how much degrade cytoplasmic material in via autophagy in yeast. So our groups still stick on the yeast cell and solving many fundamental questions. For example, I will show several examples. And one is a starvation condition. As I said, zinc is a very important ion, a cation for life. And zinc starvation induces massive degradation. And that is important for recycle of the zinc ion. And metabolic analysis of the after autophagy, you see the nucleoside transiently increase like this. And that was a massive ribosome is taken up, so RNA should be degraded via autophagy. And we found a pathway of the RNA degradation shown here. And also, Shiho Makino worked on what kind of messenger RNA is degraded by autophagy, and we found that is some sort of selectivity. Some messenger RNA are degraded, some messenger RNA are escaped from degradation. So that might be another future problem to solve. And recently we found another autophagy that is a glycogen. As you see, black structure around the cytoplasm, but glycogen granule. You can see under starvation conditions, so many glycogen is accumulated in the cytoplasm. But under starvation condition, you can see almost no glycogen within autophagic body shown here. So autophagy is glycogen is escaped from degradation by this way. But if we found one protein is a receptor of the glycophagy. If we express this gene, or glycogen is taken up, shown here. So glycogen degradation or uptake is regulated some way. And also in E cell advantages of biochemical studies is easy. We isolated the autophagic body recently, and proteomic analysis we found, shown here, some protein, autophagic machinery is preferentially taken up by red box, and some protein, glycogen-bound molecule or nuclear protein or cytoskeleton is the less preferred, almost escaped from degradation. We found one extremely degraded protein shown here, and that we analyzed this protein carefully, and N-terminal small region is important for this preferential degradation, and membrane binding alveolar helix and AIM structure is essential for this function. And if we put this small peptide to the mitochondria is taken up. So actually, this small peptide is autophagy degron. And we thought we might find this kind of protein in mammals in near future. Sorry. Okay. So, like our recent conclusion, degradation in bulk autophagy is not random process. There are so many. So, lastly, I want to say about my 35 years of autophagy research. It was just curiosity driven by basic science. I didn't expect any medical application when I started. And I had various kind of luckiness shown here. And my hope from our basic research, this is my hope, spreading the scientific way of seeing and thinking is spread over the world. And the importance of basic research and also society respect their scientific knowledge, not so much short-term usefulness, or not follow those kind of. That is very important for us. So this is my message to young generation. So I stop here, and any question or discussion, please. Thank you so much for attention.
Thank you for your very informative presentation. We'll now open the floor to questions. If you have questions, please come to the microphones. And please state your name and affiliation when you speak.
Thank you for lecture. My name is Laureate Nishiyori from Nagasaki University. So my question is, how does the isolation membranes recognize bacteria? Yeah.
Okay, bacteria do not, as I said, isolation membrane do not recognize the bacteria itself. Okay. Bacteria is taken up to the cytoplasm via endocytosis. So bacteria is end up by endosomal membrane. And endosomal membrane disruption is a signal to end up bacteria. That's a mechanism to eliminate the bacteria, invasive bacteria within cell.
Okay, thank you.
Hello, my name is Mark. I am a molecular biologist from the Philippines. Thank you very much for the very interesting talk. My question is, you've shown us several examples of situations or like effects of when autophagy becomes dysfunctional in a cell. But do we have any other examples of when, of the contrary, when impairment in the process of autophagy is actually beneficial for the cell? Like, for example, I think you've shown the role of autophagy in antigen presentation, but for instance, in the pathogenesis of diphtheria, protein degradation actually promotes the pathogenesis, since the active component of the toxin is the one that's degraded. So in this instance, autophagy may actually be, impairment in autophagy may actually be more beneficial to the cell. Or do we have any other like examples of those instances?
Yeah. Probably each system showed just basic mechanism of the membrane formation, and autophagy is survival mechanism for the cell. Once established those kind of membrane system, mammalian cell develop many kinds of physiological function to add those kind of system. Elimination of bacteria or virus, or even some specific organelle, damaged organelle. But in general, I won't say autophagy function as a survival mechanism, as a beneficial mechanism to the cell. But many people say there are too much autophagy, it kills the cell itself. That also might be true.
Thank you.
Sorry.
Thank you.
Thank you for our lecture, Professor. I'm Mike Kikumoto, a neurologist from Hiroshima University. So I'm doing research on neurological disorders and especially neurodegeneration. So I'd like to know the possibility of manipulating autophagy because one of the main causes of neurodegenerative disorders is the aggregation of some pathogenic proteins such as amyloid beta or phosphorylated tau proteins. So how can I say? So I'd like to know if we can use or manipulate the autophagy system to eliminate these coagulations.
I'm not an expert on that kind of subject, so okay. As you know, there are some protein. We thought that the cytoplasmic protein is reside individually, but these days some sort of the complex formation, further phase separation might be very important. And so such kind of the state of the protein is recognized by the autophagic machinery. That's also made by phase separation. Those kind of understand now become popular. And as I just said, the glycogen desires so much, but it's escaped from the lineup. That's one example. But we didn't know why about one protein is expressed, glycogen become a target of the autophagy machinery. So we need such kind of biochemical study or more even biophysical study on the state of the protein in the cytoplasm is very important question in your future, I think.
Thank you for answers.
Hi, Professor. Thanks for your talk. I'm Meng Lan from Singapore. I wonder, is there anything similar to autophagic lysosome reformation in the yeast? For example, after because there are more membrane fields to the vacuum during the autophagy, but after the autophagy finish, the vacuum has to return to its original size. So there are extra like membrane that need to go somewhere. So like, what's your understanding about that process?
That's a good question to be answered in future. Well, for example, many autophagosome is fused to vacuole. So membrane should expand, vacuolar membrane should be expanded, but it's not true. So there must be some mechanism to membrane flow from vacuolar membrane to somewhere, but not much. Even in this case, we don't have such kind of analysis yet.
Thank you.
Any questions? Okay.
Thank you, sir, for your nice presentation. Sir, I'm Dr. Mahabub from CSIR Bangladesh. So hopefully you visit Bangladesh coming month. So I have a very simple question. So fasting is one of the way for autophagy. So how long time fasting you recommend for autophagy?
Yeah, I often, when I go abroad, I have so many questions like you said. But I'm not an expert on the problem. Probably fasting might be activate the autophagy, so it's quite reasonable. Good fasting might be helpful, but it's also depend on the individual. So we cannot say. I cannot say what is the best way to take a meal every day. So I have no idea. Sorry.
Thank you, sir.
Yeah.
Okay, we have run out of time, so I guess we'll finish here. Thank you, Dr. OHSUMI. So please give a big round of applause to the Professor OHSUMI for the last time.
Thank you very much, Dr. OHSUMI and Dr. Fujioka. Let us again show our appreciation with a round of applause.